You guys are good, yes. Because the peptide bonds are between the amino acids. If you think about objects, there's always one fewer betweens than there are objects that you're talking about. Okay, well, a linked together group of amino acids like you see here is sometimes referred to as a polypeptide chain because it has a bunch of these polypeptide bonds linking the amino acids together, or sometimes it's just called a polypeptide for short, or some people would just shorten it even more and just call it a peptide. Several amino acids linked together by peptide bonds. You can talk about the backbone of a polypeptide, and that just means the backbones of all of the amino acids. The backbone of a polypeptide is amine group, central carbon, carboxylic acid, carbon, amine group, central carbon, carboxylic acid, amine group, central carbon, carboxylic acid, carbon, all the way down the chain like that. In other words, the backbone is the whole thing except for the R groups. The R groups do not count as being part of the backbone. Notice that no matter how many amino acids you have in your polypeptide, the first amino acid that you start off with will always have its amine group hanging out there. And the last amino acid that you added on will always have its carboxylic acid functional group kind of dangling out there like that. And so you can talk about the amine end of a polypeptide, that's the starting end. And you can talk about the carboxylic acid end of a polypeptide, that's the finishing end where the last amino acid got added. And like I said, the average protein is 400 amino acids long. Some of them are thousands of amino acids long. The shortest one I ever heard of was 8 amino acid, but they might even come smaller than that. And just to give you a sort of a math question here. Yeah, this will test how well you do math in your head. How many possible 3 amino acid proteins are there? Let's see who's good at math in their head. If I said how many possible 3 amino acid proteins could be made? What'd you get? 27 now. Well, how many possible amino acids could be the first amino acid? 20. How many could be the second? Third? 20. And so mathematically you have a certain possibility and then another possibility and then another possibility. You multiply the possibility, so it's 20 times 20 times 20, which I think is 8,000 if I'm doing the math in my head. And that's just for a 3 amino acid polypeptide. If you consider that the average protein is 400 amino acids long, and you have 20 possible amino acids in each of those slots, you get this astronomical potential number of different proteins that can be formed, which probably is why proteins are used so much in cells. Remember, they're the enzymes, the transport proteins, the receptors, because there are so many different proteins that you can form, and they can fold in so many different ways. You can just imagine any type of protein to do any job you can think of could be formed. There's so many possibilities. Now, just a moment ago, I told you that when you link a bunch of amino acids together, it's called a polypeptide or a polypeptide chain. But a few minutes before that, I told you a protein was a bunch of amino acids linked together. So you may wonder, well, what's the difference between a polypeptide chain and a protein? And they're sort of the same, but they're sort of different also. To explain it, back to my Skittles view. To explain the difference, I like to show this slide here. Think of kitchen implements, things you use in your kitchen. Some things you use in your kitchen are just a single object, like a pot is just one piece of metal, and it's able to carry out its function of being a pot. But other things in your kitchen require 2 things to click together, like your blender has a pitcher part and a motor part, and until they click together, it hasn't become functional. Well, proteins are chains of amino acids that carry out some sort of function in the cell. Some proteins are able to function by just being a single polypeptide chain. But other proteins don't become functional until two polypeptide chains have clicked together with each other. They're more like a blender. And so when we say a protein, we mean either one polypeptide chain or several polypeptide chains clicked together that are functional, that are able to carry out a function. And it's not limited to two polypeptide chains. Some proteins require three polypeptide chains before they become functional, and some proteins are four polypeptide chains coming together before they get functional, and that's probably not even the upper limit. I'm sure there's some that have even more, require even more polypeptide chains coming together. All right, now let's talk protein folding. I think I mentioned at some point that proteins have a natural tendency to fold themselves up. They're like old rubber bands or old pieces of fishing line. If you pull a piece of fishing line or a rubber band out straight, you can pull it straight, but if you let go, it has a natural tendency to wad itself up into a highly folded structure. Same thing with proteins. And that folding is important, and it's good because remember the binding site, or the active site if you want to call it, talk about an enzyme, the part of the protein where it binds onto the molecule it's supposed to be working with only forms when the protein folds correctly. So a lot of research and studies have been done to learn about how proteins fold themselves up. And there's some, they call the folding of the protein or the shape of the protein, they call that the structure of the protein. And the people who study this subdivide the folding, the structure, into four different levels: primary structure, secondary structure, tertiary, and quaternary structure. And let's look at these things. The primary structure means the sequence of amino acids in the polypeptide chain. In other words, just what order the amino acids are in the polypeptide chain. So imagine if you had a protein and you wanted to find out its primary structure. If you could magically shrink yourself down and grab a protein with your hands, you would pull it straight and you would say, okay, Mr. Edens, the primary structure is, and you take a deep breath, you'd say methionine, proline, glycine. You're just naming the amino acids in the order that you find them: tyrosine, valine, isoleucine, glutamate, aspartic acid. You would always start off with the terminal amino acid, because remember, that's the starting one. That was the first one in the chain. Anyway, you work your way through. Cysteine, proline, glycine, tyrosine, alanine, serine, alanine, glycine, aspartic acid, until you finally get to the carboxylic acid, and then that's the end of the primary structure. Now, primary structure is important because it controls the overall folding of the protein, and the folding of the protein, remember, is what forms the binding site. And so think of the primary structure as the programming that's going to tell the protein how to fold itself correctly. And even if you were to somehow change, to mutate just one of these amino acids, even just one change is often enough to cause the entire protein to misfold, and then it's not going to be functional, or it probably won't be functional because you probably won't get the right binding site forming. Here's an example of just that. This is the protein hemoglobin, which is the one inside your red blood cells that carries oxygen for you. Hemoglobin is actually made out of four different polypeptide chains that click together, and they showed light blue, dark blue, and yellow and green right here. These are actually some coenzymes that help bind the oxygen. But anyway, hemoglobin is made out of four polypeptide chains. And some people have a mutation. There's a disease called sickle cell anemia, where some people have a mutation in just one of the amino acids in one of the polypeptide chains. Here's a diagram of it. This particular polypeptide chain is 146 amino acids long. And here's the first 7 amino acids of the 146. So imagine this would keep going or 146 more amino acids. That's normal hemoglobin. It has a glutamate at position 6. In people with sickle cell anemia, that 6th amino acid of the 146 has been mutated to valine. Here are the two R groups being compared. You can see that glutamate is an ionic R group, whereas valine is nonpolar. And so that's enough, that single amino acid change is enough to misfold the whole protein. And so people with sickle cell anemia, their red blood cells, which is where you find the hemoglobin protein, take on a bizarre shape because the proteins inside them are in this non-standard shape. And this ends up giving them problems carrying oxygen in their blood, and that's what anemia is when you're weak because your blood doesn't carry enough oxygen. All right, so primary structure, just the sequence of the amino acids, and it's important because it dictates all the other folding levels that follow. Okay, imagine, so we had our protein pulled straight like this. Imagine that you didn't let go of the ends, but you brought them just a little bit closer together. What you'd see is the protein would start to fold itself in some certain patterns. And those patterns that the protein starts to fold itself into are called the secondary structure of the protein. And it's defined like this. They are areas of regular folding of the polypeptide chain. And by regular, they don't mean boring and plain. They just mean patterned parts of it that fold into a recognizable pattern. There's actually only two patterns that you see in secondary structure. One is called the alpha helix. It's a corkscrew-shaped folding. And the other is called a beta sheet, which is sort of a zigzag back and forth shape of secondary structure. Both of these secondary structures are caused by hydrogen bonds along the backbone of the polypeptide chain. Here's a close-up of the alpha helix. The ribbon represents the backbone, but they've drawn in some of the amine groups and central carbons and carboxylic acid carbons along the backbone. And as you can see, you have partially negative oxygens there, partially positive hydrogens there, and because they want a hydrogen bond to each other, they arrange themselves in this alpha helix shape because that allows them to hydrogen